So I'm Nancy Simonian. I'm the CEO of Syros, which Rick introduced to you, which Rick was the founder with Jay and Nat. My background is I'm a physician by training. I was at Mass General Hospital, trained as a neurologist, and just by virtue of the, 20 years ago when Phil had started Biogen and they came across the first drug to slow disability progression and didn't have a neurologist, that was really my opportunity to move from academic medicine into biotech, which I've been in for the last 20 years. Just about 3.5 years ago, I had the opportunity to meet Rick and a couple of the venture capitalists and decided that this was a really amazing opportunity to work in a disease area or a platform that could fundamentally change the way we think about human disease, and really was the basis by me joining the company. And just to give you a little bit more context, at Biogen I worked on 2 really important drugs in multiple sclerosis, interferon beta and then an anti-adhesion molecule called Tysabri, and they made it to the market. They were impacting a lot of patients, and then I was at Millennium for 10 years and I was their chief medical officer there, and I brought to market Velcade, which is a drug for heme malignancies, and Entyvio for inflammatory bowel disease. So I actually had the opportunity to see molecules move through development onto the market and help patients. But in all of those cases, those were molecules looking for patient populations. They all started with a group of people saying, I want to work on this target or this pathway, and then try to figure out where they go. And while those were successful programs, most of what we did both at Biogen and Millennium, and I argue across the whole pharmaceutical industry, most of it doesn't work. The failure rate of, the 90% things fail that go into the clinic, that's true. And I think it's largely because we don't understand fundamental disease biology. And what got me so excited about Syros, at the time it was called LS22, and I was in Rick's office, was the opportunity to sort of say, could we use this understanding of gene regulation to understand the fundamental biology around why a disease cell is no longer a normal state? Why is a cancer cell in a different cell state than a normal cell? Can we use this lens to understand that? And so that was really what got me excited to embark on sort of this, starting up a company from scratch with the scientific founders and the VCs.
And so I just want to point out, the inception of this company was just over 3 years ago, 2 INDs this year. That's extraordinary, Nancy. Congratulations.
Well, I think it's, as Rick said, we just had our first notice of our IND clearance on Friday of last week. And what's really exciting was just a year into the company, by analyzing these enhancer regions of patient samples. So we started the company by saying, let's not focus in cell lines or model systems. Let's start directly in human disease tissue. And we brought in, and through collaborations, initially studying this in acute leukemias and in breast cancer, we analyzed these enhancer regions, identified that there were subsets of patients in both breast cancer and AML that had super enhancers controlling a gene, a retinoic acid receptor alpha gene. And from that discovery, then we identified that there was already an existing molecule on the market in Japan for a different indication that actually worked in patient models in the laboratory that had that biomarker. And we brought in that drug and now are poised to go right into a phase 2 clinical trial in a biomarker selected patient population based on this understanding of this novel dependency. So I think one of the reasons we've been able to move so quickly is because this insight and this lens is, I think, really producing a high yield in terms of disease dependencies. So we're excited about that. And of course, we have our second program, which is targeting a kinase that's part of the enhancer complex that we think is important, CDK7, in controlling the level of expression of important oncogenes. So I think we were fortunate at the foundation of the company, and really give credit to Rick and Nat and Jay, who brought to the table at the beginning a combination of the biology, chemistry, and translational piece. So that was always at the foundation. We weren't just a science experiment. And one of the reasons I loved the platform and all, I'm a physician and I wanted to be able to help patients. I didn't want this to be a tenure long? Can we figure out the rules? I wanted to say, can we take some low-hanging fruit and bring it forward? And we're really excited about that opportunity.
So I'm sure there's some questions. We'd be happy to entertain them. Go ahead, please.
I think I heard you say that you used biomarkers to select the patients. So can you elaborate on that? Because sometimes it's hard to have specific tests, or biomarkers can vary. So can you elaborate on that, please?
Yeah, sure. So, we made the observation in patient tissue that if there was a super enhancer controlling this gene, it turned out that that was denoting tumors that were highly dependent on that gene, that retinoic acid receptor alpha. And then we developed a biomarker that's associated with the super enhancer. So one of the things the super enhancer does is controls higher level of expression. So we're able to use and develop a biomarker related to the super enhancer. We haven't disclosed the biomarker. It's actually part of our sort of, we have proprietary insight and a filed IP on that. But I'll just say that it's what's been great is we're now developing biomarkers that are highly correlated with the enhancers that we can measure in peripheral blood from patients with these cancers.
There's another question over here. Yeah. We'll get another one for you. Maybe we could go ahead with your question, and we'll come back.
Thank you very much. The AMD project, fibroblast RP program. My name is Tina Chu, and I'm an ophthalmology physician by training, interested in biology. There has been a long time, I think, people was arguing the IPS, are they behave as good as pluripotent embryonic stem cell. One of the key is mitochondria. So through reprogramming, I wonder if you could use your transcription factor to predict if those cell aging process are the same as the one you generate, and how old, how young they are, and the pigment granule within RP derived from the programming. I think that that's a key issue for how we predict those cells behave after transplant. I know Takahashi's work very well indeed, did a lot of research on that area, but I don't know the answer whether or not we know when to transplant those cells. We know, okay, roughly how many days of passage and blah, blah, but do we have a biomarker, say, this is the best timing to put them in the eyeball, in vivo, and then how soon those cells may, are they aging faster? Because RP aging is the primary driven force or root cause of age-related macular degeneration. If the grafted cells are already at their mid-50s or something, you don't know, but the key is, can you comment whether or not there are any transcription factor could help identify those cells for suitable time?
Right. So just to be clear, partly because of all those clinical issues, we're still working on that in an academic setting, and it's not at the moment one of the programs within the company. But we're trying to work with folks in Kyoto to just understand the fundamental biology. I actually think it's pretty remarkable. We've been able to take human cells and turn them into what look like functional RPs, but it doesn't necessarily mean they're ready for clinical translation. And one of the reasons is exactly what you mentioned. We don't know all the properties of these cells. We don't know, for example, maybe if we derive them from blood cells, they would be younger in their sort of physiological behavior. So we've got a ways to go to understand them before we'd want to attempt to translate that.
Yeah, I think there are some research, some data show the reprogramming, the DNA, of course, they are younger. And they also have some influence to the mitochondria. Means if you look at the entire genome and all the growth factor and those molecules and the markers, they are quite similar, but it's not exactly reached the level of embryonic stem cell, so the iPS. We know that, but we don't, as you mentioned, pointed out. They will actually, she was here last year at the ALCON MEI meeting, presented the data. It was fantastic. I want to comment. Thank you so very much for bringing that.
You're welcome.
Thank you.
You're welcome. Yes,
Hi,
please.
I'm Vincent Ling at Takeda Pharmaceuticals. We actually work with Yamanaka on many regenerative medicine projects. So I'm very interested to hear about you talking to Yamanaka to see how he can incorporate your discoveries into his programs. That's very exciting indeed. I just want to remark and maybe get your opinion. I remember about 15, 20 years ago, I remember I was at Harvard then, and Irv Weissman gave a talk on how he insists that transdifferentiation is impossible to do. And it seems that you've broken through that paradigm. And I was wondering, do you know if thoughts in the field, are slowly starting to change, or do you think, there's still some levels of resistance to overcome? I mean, it's really interesting because at first people didn't think you could make ES cells from humans, and then couldn't make iPS cells, and now those barriers have been breached. But to me, I think the really significant finding is if you can go directly from one cell type to another and truly transdifferentiate.
Yeah, this is a little like the thinking 15 years ago that you couldn't selectively drug kinases, and then you can, and you can't drug transcription, but I'm advocating that you can. So just to be clear for people who don't appreciate this, reprogramming is the process of taking a somatic cell and turning it into a pluripotent cell, something that would be akin to an embryonic stem cell. Transdifferentiation is taking a differentiated cell and turning it into another differentiated cell without going through that early embryonic program. And so it's only really been in the last couple of years that we've appreciated that we can do that. And there are a number of instances where that's been demonstrated.
That's very exciting.
Yeah.
I'm Howard Fingert from the Department of Internal and External Innovation at Takeda. Nancy, having had the absolute pleasure working with you and knowing the breadth of your experience with leading companies, evaluating companies, I have more of a question about the topic of convergence that this whole program's about. And as I see what you're doing now is you're sort of sharing with us the Syros story, which was a successful result of helping to lead a path from the convergence, the integration of you mentioned the venture capital, the financial side, the science side, the leadership, the clinical, towards a company, a new company, that hopefully will make sustained contributions. This is not going to be a pump and dump, right?
Correct.
You want to be here 20 years and 40 years from now doing more of the same work. So I'd like to know if you could share with us, what do you view as being, thinking back the last 3 years, what are the best working practices that you'd advise to others about what are the elements or working practices that you think are the highest priority for success in the same kind of enterprise going forward? Because we want convergence to lead to success,
Right.
and not because there have been convergences that have led to failures.
Right.
So
Right.
can you share your thoughts?
Yeah, sure. I would say a couple things. If you said what are the top 10 priorities that I've had since I started the company, I'd say like 1 through 9 is around culture and people. And that's really what I spend my time on. How do I make sure that we get the right people in the company? How do we ensure that we create the right environment to allow innovation and risk-taking at the same time of ensuring that we actually are on the path to moving things along and creating value and all of that. I'd say that has been a huge piece for me in terms of the importance of what it takes to build a great company. I mean, we have an amazing scientific platform and amazing scientific founders and board of directors, but at the end of the day, it's really about the people and the culture that you're building there. So I'd say that's, like I said, what I spend most of my time on. The other is really around the importance of financing. and how to use this community here in terms of all the relationships and to try to leverage that to help to bring in the dollars that it takes. I mean, as you all know, developing drugs, well, identifying, identifying targets and then developing drugs is a very, very costly business, and it just takes continual time, raising money. So how do we leverage the community? What's been so great here is that when I decided to do this, I was at Takeda, I ran a very large organization there, but I'd never been in the situation where I had to go out and raise money for myself. The great thing is that I had several colleagues over the last 20 years that had moved into the role of being a CEO. And so they were able to introduce me early on to a lot of their investors, public company investors, and I just started my networking 3 years ago, meeting with people, introducing the concept. Of course, this area of science is incredibly exciting, but it's also kind of a new thing for a lot of people that spend their time thinking about other areas in science. A lot of times on education, getting to know me, Rick was involved in some of those discussions. So I'd say sort of leveraging the internal network of people in the area to create the right relationships, because this is, at the end of the day, also a relationship business. And that people have to know you, trust you, deliver. It's also how you attract and retain great people too. So those would be the two things I'd say, Howard.
All right, I think we'll have 2 more questions. Go ahead.
Just returning briefly to the science, Steve Gullins. I'm curious about how this process of the enhantiome is going to play out in the tumor field in particular. Is it going to be more like a targeted therapy where each tumor has a specific enhantiome that's going awry? Or is it going to be like the immuno-oncology field where one drug may hit 80% of all tumors? Have you done a survey across tumors to see, are there general principles? Is this 100 things we're trying to target or 2 or 3?
Maybe I can start, then I'd see.
Yeah.
We've analyzed, we probably now have the largest database of human tumors that we've analyzed these regions. And we certainly see, and we'll just use MYC as an example. I mean, MYC always has, one of the largest super enhancers across many different tumor types. So we do certainly see genes like MYC that we see common, but we also see other things that are, in subsets of patients, but we actually see them across. So as an example of the RAR alpha, we made the discovery initially in our breast cancer tumors. We then went over to AML, noticed that it was in a subset of AML patients, and we know just through other publicly available databases that there are subsets of other tumors. And it makes a lot of sense because it's important in sort of de-differentiation or differentiation of cells. So it's just one of a common pathway that cells use to be in a de-differentiated cell state. So I think it will be a combination of things. I don't think it will be some that are more common and others that are more tissue specific. But that's our experience in the human tumors. And
yeah, remarkably, the tumors are telling us through these super enhancers what they're most dependent on. And the hypothesis here is that it's not just one, but they're developing multiple dependencies. A drug like Nancy's CDK7 drug might simultaneously hit multiple dependencies, and the hope then would be we'd run into less resistance, and that it would be a more broadly active drug across many cancers. But I think the history of this experience is that it'll be all of the above. There'll be some ways to target a broad spectrum of cancers, but there will always be those cancers that require special attention.
One last question.
Hi, my name is Belén Carrillo Rivas. I lead Global Clinical Submissions Quality at Pfizer. So the question that I have for you, we're talking about disease heterogeneity and tumor heterogeneity. So how do you translate the knowledge that we have on tumor molecular phenotypes into meaningful clinical trials that are useful for patients?
well, I think at least the way that we're approaching it is, like I said, start initially all of your work in human tumor tissue. So in the case of AML, we got, through an academic collaboration, got 80 or 90 AML patient tumor samples that then we analyzed. So we already were, and that was from the peripheral blood. So then when we go into the clinic, we're actually basically taking those same cells to identify the biomarker that we're looking for. So trying to get initially in your discovery work as close as possible to the actual disease that you're studying. It's certainly a lot easier when you're dealing with the hematologic malignancies, but that's the approach that we're taking.
So I guess my last question would be, you have to have a validated biomarker, right?
Yes.
So
Yeah.
it's about, sometimes the complexity of those clinical trials is that you need to validate the marker at the same time you need to validate your therapeutics. But I guess, that's where many people see precision medicine being kind of complex, because it's not just about validating the therapy, also validating the biomarker for that specific tumor. I guess that's where my question was going.
Yeah, no, absolutely. No, so it's a great question. And, there is quite a complex process that needs to go through just through the assay validation, but then there's also kind of the validation in terms of what the right cutoff level is, and that's sort of a clinical validation piece. So there's definitely many steps. It's almost a parallel process to development of the drug, and it's quite labor intensive, but it's doable. And of course, we have in cancer multiple examples now of targeted drugs with companion diagnostics. So at least there's a path forward, and we have the FDA working well with CDRH to ensure that we can actually do this much more seamlessly than in the past. So I think there's a good path, and the FDA is very open and welcome to trying to make this more of a reality for more therapies.
Andy, did you have some?
Yeah, so it's just one last question in preparation for our next speaker. We have a number of young researchers in the audience. So for Rick and Nancy, can you give some advice to younger faculty thinking about going down a commercialization pathway? What would your advice be about how to go about it, and just words of wisdom that they can take and make use of?
Well, I'll start this one, and I'll say, I'm going to paraphrase Nancy. There are three key ingredients: people, people, and people. And the first thing Nancy did after she became the CEO was to steal all the top people in my lab.
But he actually
She
allowed
literally
it, so
did
I'm
that.
just telling you.
I've spent the last 3 years rebuilding my laboratory. So one of the things that I have found so special about this community, and is the reason I've been here for 30 years and have not left, is because I can't find another community where really, really good people exist in all the scientific, business, financial, legal spaces within walking distance.
So I think what I would add to that, I was on the academic side and then I went into biotech. I think it's a very personal decision. I think you just have to decide what is it that you really like doing. And so, for me it was, I wanted to be able to really see, the scientific work I was doing translated into helping patients. And so if that's really your drive, I think going into industry is just a wonderful thing to do that. But I also say that, we absolutely rely on, it's the foundation of what we do is based on all of the work going on in academic research. So they both have to coexist. And I think for each individual determining what they want to do and is whether they want to be on the inside or they want to collaborate. I just think there's so many opportunities to be able to identify ways to translate your work into helping patients. So that's what is important to you.
Thank you so much. Please join me in thanking Rick and Nancy.